Okay, let's get started. Let me introduce you to our panel, which is most probably not needed, but anyway, we have one participant connected to us online, Frans Verstraete from Brussels. Good to have you. I'm looking at the monitor, but there you are. Welcome. And we have another 5 joining me here in Berlin. We have Nicolaus Kries, Head of the Department Risk Assessment Services at the EFSA. Then we have Claudio Carlon, Head of the Unit Biocidal Active Substances at the ECHA. We do have Hans-Jörg Matus, Head Genome Safety at the Novartis Institutes for Biomedical Research. We have Eugenia Dogliotti, Professor Emerita at the Istituto Superiore di Sanità in Rome, renowned for several multidisciplinary studies on genetics and cell biology. And we have Professor Tanja Schwedle, Vice President of the German Federal Institute for Risk Assessment. A warm welcome to all of you here in Berlin, and a warm welcome to Brussels as well. Okay, now, we've got quite some thing behind our back already, or at least you do, since you are here since Monday. And as a start, I would like to know your personal point of view. How was, let me, sorry?
I can't hear you right now. They are on it. Just that you know, if you talk to him right now, he can't hear you.
Okay, I know. I don't know whether you got that as well. It might be important. Frans Verstraete can't hear us yet, but they're working on it. So I hope.
Now I can hear. Now I can hear.
Oh, perfect. And we can hear you too. A warm welcome to Brussels. Good to have you.
Yeah.
Okay. Where was I? I wanted to start with your kind of conclusion of where we are now. So if you reconsider all these things that you heard during the last 2 days, which approach seems to you personally the best to pursue? Is there anything that you would say like, yes, that's the thing. If I was king of Genotox, that's what I would do.
Thank you, if you allow me to start. If it was that simple, we would have done it on Monday morning. And wouldn't have had 2 days of experts meeting in 1.5 days of intensive workshops, sometimes till 9 o'clock. Congratulations to the colleagues, it shows the enthusiasm. And thank you very much, by the way, for BFR for getting everybody together. And therefore, I know you want a short answer, but if the short answer was there, look, we have Paul White from Health Canada coming and experts from all over the place, we would have been able to tell you that.
But is there something that you personally find most promising?
Of course. Personally, I would find NUMS most promising. And yes, obviously, quantitative, very promising. That's also personally, because I should put a disclaimer here, I'm a vet. I'm not a toxicologist. And also, as a vet, I have to say I'm very committed to the 3R principles, and we should all be committed as a society to those principles, and they mean going away from in vivo testing, going to in vitro and quantitative testing. We're just not there yet, and that's what we definitely heard. If you allow me one last remark, because what I heard a lot was paradigm shift, was one, and decade or 10 years. And it reminded me of a story from the old Soviet Union where a man goes to buy a car and they say, yeah, it will be 10 years till you get it. He says, morning or afternoon? And they say, why? What difference does it make? And he says, in the afternoon, the plumber comes. So if I hear 10 years, I hear we're kicking it into the long grass. And I think we're also here to try and make sure we maybe get it within the 10 years.
Okay, we can end this discussion by making it within 10 months, maybe. But first of all, I would like to ask you, what was your most appealing moment? What was the idea that caught you?
Thank you very much for asking, and happy to be here. Thank you for hosting me. I'm not a genotoxicologist by education. I learned a lot from my expert in here in these 2 days. I was impressed by what has been discussed and the tools and the advancement in science. the learning is that a question remains. What do we want to achieve? Because indeed, we have been talking about shift of approach, paradigm shift. And I would like to turn the question from the perspective of the regulator. What do we want to achieve? A paradigm shift for what? And the question could be, okay, do we want a paradigm shift because we want to move from a system where we want to prevent exposure to mutagenic substances to a system where mutagenic substance should be considered, and genotoxic substance more in general should be considered in terms of potency and regulated as such, or we want to have a safety net for those substances that, for example, we cannot replace, or we have good reason for not replacing, and we want to apply these methodologies to see whether there are conditions, there is the margin for that. And if we want that, the second, I think that there is already a margin of application in the current regulatory framework. We have already some experience, at least in the definition of exposure risk curves that is provided to risk managers to see what is the cost benefit in relation to risk delta. And is it enough? Is it useful to have a definition of safety threshold? And under which condition? Is a safety threshold based on very conservative assumption? So potentially even very, very low, maybe even counterproductive. So the question that I come back home with is to understand how the current qualitative approach, which is very much centered on the classification, and qualitative approach can be combined. And what is the perspective in future? It seems to me that there are advancements. As Nick said, there is also quite a lot of promising from new approaches. Methodologies can complement information that we don't have at the moment. And there is margin whenever we see that a substance, even for regulatory substance might not be approved, but there are good reasons to reconsider the potency to see how these apply. So for me, it's not a yes or no. It is how to move on.
Yeah, exactly.
And how a combination of the two approach in certain cases can pave the way for the future. And for that, I go back home with quite a lot of optimism.
Well, you said you take home a question, but do you also take home a possible solution? Something that you would say, like, well, there are like 5 paths, and I would go that one?
Yeah, I didn't want to be understood as a person questioning only. For me, a combination is a solution. So I see the possibility, and I see that under certain circumstances, we can already see how it works. So what has been discussed today, yesterday, and the day before, was quite a comprehensive review where we are. And it seems that we have a number of instruments that still do not clarify totally the uncertainty we are dealing with, but there are tools that we can use. The question is where the tool can be used under the current regulatory framework. And I can see that there is the possibility.
Okay, thank you very much. So, Hans-Jörg Matus, which approach seems most appealing to you?
Well, if there's one thing, I mean, we've spent now the last, 2 days, 1.5 days, reflecting back and forth and back again on the question whether we can safely use genotoxic compounds, essentially, right? That was the core of what we talked about and what we thought about. And quite frankly, I mean, we didn't invent new data. There was no data that went out there, and we were all aware of that. But after that quite intense discussion, I think we're just ready for use quantitative data, benchmark modeling, these type of things in a regulatory field. And I think we should move on there in a formalistic way. I mean, we have whatever. I'm coming from drug research, so you have your approach. You screen very early, and you throw out the bad guys, the bad molecules that are genotoxic. You avoid them altogether. So there is a way we can work with that. It's not very painful, but of course it's a question: could we tap into new chemical space that we can't use so far in a safe way? And then in other situations, in other regulations, in the environment, in what have you, in food, there are exposures that are not so easy to avoid, like if you develop a new molecule. And I think we are there. I think we should try to get that now in a formal framework, in a whatever I say, OECD or something like that, and really define what would be needed. And because if you start such a process, you would also define what is still missing to get to an acceptable validation of BMD approaches, right? So they can be used then in a regulatory field, in a in this question how to use genotoxic compounds in a safe way. That's my main take home, I have to say.
Okay, thank you very much, Professor Rigliotti.
Okay, so I have to say, I have been working for 30 years in the field of genetic toxicology, addressing the mechanisms of genotoxic compounds. So I was really positively surprised seeing that the statement to deepen the mechanistic understanding the mode of action of genotoxic agent was appearing on the slide of almost every workshop that was just, attended by different people in the past days. So if mechanistic insights are important, what should we do? So I think that as first, and this is a very short-term type of goal, we should really have a semantic approach. And when I'm saying this, what I want to say is that we should also abolish a certain type of terminology that is, I would say, outdated. Because up to now, we are doing a sort of qualitative assessment of genotoxicity data. And we have this famous binary classification: genotoxic, not genotoxic, DNA reactive, non-DNA reactive, indirect, direct, threshold or not thresholded. This, I think, is very confusing. We should really abolish this terminology because, indeed, the mode of action tells us that this terminology doesn't really apply clearly to what the mode of action of genotoxic agents is. And I think the first day, Paul White gave this very interesting talk about this thresholded type of dose-response curve for mutation. This was for mutation, and the case study, I think, was ethyl methasulfonate. So, of course, my question as a DNA repair person, I immediately said, okay, we should investigate how many DNA repair pathways are activated in that area of low doses. And indeed, this is not trivial because we have at least 3 or 4 different repair mechanisms handling methylation damage. So it's really a multi-affair task for DNA repair to handle that damage. So it's a very complex situation. And then if we go to arsenic, and I think that we have been working on that for more than 1 year, right, in the scientific opinion. That one, also, you say, okay, it should be thresholded because it's indirect, but it does inhibit DNA repair. So this is going to be a big problem. It's really a special genotoxin. So I don't want to go into detail because only myself is very interested in this detailed mechanistic aspect. But what I want to say is that we should be really flexible. So I wouldn't really put any gauge when we are running a mode of action analysis. We should look at data, and we should also improve the tests that would allow us to really open a window in this very long process that goes from the stressor to the outcome. And saying that as approach, I think that this is a long-term one. I think that the integration of the adverse outcome pathway with genotoxicity assay is going to be extremely interesting because they identify initiated events and key events along the process that go from the stressor to the adverse health outcome. So it's a fantastic palestra, I would say, for people who are working in genotoxicity, is to measure, to try to measure these key events. And so to superimpose the two approaches, I think that is a promising goal for the future.
Thank you very much. Before we pass on to you, I would like to give the baton to Brussels. Frans Verstraeten, I don't know how many of the talks you could join, but anyway, from your personal view, what do you think is needed and what is most promising to develop new strategies?
Well, what I indeed also already indicated, I think in the workshop on Monday, is that what I hear the discussions is sometimes they say, yeah, but there is indeed, there is the impediment for further developments because of the regulatory requirements, which are indeed not allowing to change the approach or something like this. And that, my answer to that is, and I also discussed it even with my colleagues here in Brussels yesterday afternoon, when indeed there is a scientific consensus for an improved approach as regards genotoxicity, then of course we can always adapt legislation to make that possible. So this is, I think, the main message that I would give is that indeed, if you were looking into better ways to assess genotoxicity, that you would not have to say, yeah, but we cannot go that way because there are these and these regulatory requirements. I would say, well, come forward with a scientific consensus on a better way forward for assessing genotoxicity, and then we will take our responsibility as regulators to adapt the regulation accordingly. Thank you.
When you are demanding a scientific consensus, and you just mentioned that you talked to your colleagues in Brussels yesterday, is there a consensus among you that there needs to be something done?
Well, if there is, I would say we are always, even if that might not be always sound like this, we are always open for indeed improved ways to ensure public health protection. And indeed, if there is indeed better ways and to better assess genotoxicity, I would not see how we can justify that legislation would block that better ways to assess genotoxicity. Importance is, of course, and that I must also admit, the regulators, certain at least, not all, but I know colleagues which might have a more conservative approach. And from the moment there is indeed no consensus in the scientific world about a certain approach, then indeed there might also be the reluctance from the regulators to change. Then they might be a bit more having a bit of fear and then say, okay, let us keep it like it was instead of changing and then facing criticism from certain scientists or from certain organizations, which might indicate that indeed we lower the protection or something like this. Thank you.
Thank you very much. So, Tanja Schwedler, taking into account all the information and discussion you experienced during the last 2.5 days, what sounds to you as a promising approach to get a step forward?
Well, I'm not 100% sure about the promising approach. So when we started here on Monday, personally, I was convinced already that we should go to the direction of a quantitative approach. What I heard over the last 1.5 days is that there is consensus that this could be helpful. I learned that we have the toolbox in principle to do so. I heard that risk management is interested, that we work on that. I completely understand and this is the basis for each risk assessment, that it has to be scientifically based. I'm not convinced that we have to have scientific consensus, because this is what I learned as a toxicologist and risk assessor: you never get scientific consensus. It should be scientifically based. That's important. Where to go to, it's really hard. We should not forget to answer some of the questions that popped up today before we start. So I heard today the question on, is mutagenicity itself a hazard? So as a regulator, I would name, can we use mutagenicity data as such as marker? So I'm not sure. We have not answered this fully yet. We have to do this before, well, in the regulatory world of risk assessment, not in the field of genotoxicity. So we have to be sure about this. If we are not sure about this, we cannot use genotoxicity or quantitative approach.
So let's jump in there and open up the discussion. What's the answer to this question? Anybody wants to answer or comment on this? Yes?
Yes, as I said, why I say it's not a yes or no. I know how the regulation is made at the moment, but I give you an example. In biocide, the mutagenicity classification would go for non-approval. Nevertheless, you can consider whether you meet derogation criteria. Derogation criteria could be that the societal impact overweight the risk. Then the risk manager needs to know a level of risk. What is the level of risk we are dealing with? And it is where more quantitative approach come to help. But you don't need to have an acceptable level or tolerable level. But all the techniques that we have been discussing today are the ones that build up the case. So there are space already in the regulatory framework where this technique have been applied to a certain extent. So it is not that there is no room, but there is a sort of room where things can start developing, and there have been some experience that have been borrowed. So I agree. We are not ready for a paradigm shift, in my view, considering all the uncertainty and the debate in the scientific level. And we are not ready to change, deviate from the current regulatory framework. I think that still work, but I see that there is the room for applying some of the elements, some of the tools that we have been discussing these days.
Yes?
I think I wouldn't know why we wouldn't use an increase in mutation frequency or mutant frequency or whatever as an endpoint to assess, or to claim this is adverse. Of course, I mean, what we are lacking is the usual definition of adverse, which is a functional defect or something like that, right? A deficiency in something that, whatever. What we have when we look at histopathology, at other things, and define this is adverse and this is not yet adverse. Okay, nevertheless, I think we have agreement to say, listen, if we have an increase, a relevant increase in mutations, this can lead to downstream adverse event, no matter what that is. So, and we can measure that very nicely. Actually, we can measure mutation frequency increase, and we can, do, for example, this quantitative assessment, and then we can define something. We can define and say, listen, whenever we go, we have a response that we can measure, that we can define. This should be considered adverse because of that propensity to induce something downstream. So it's actually a tool, even if we say a mutation is per se not, or an increase is not per se adverse, because whatever, the third base or the 97% of our genome which isn't transcribed and all these things. Nevertheless, it's a super biomarker to say something bad is going on, right? And then we can talk about the level, and we can do whatever, BMD modeling and define whatever, critical effect size, these type of things, and all that, what we've seen. Beautiful, to my mind.
Okay, thank you. You want to answer?
I agree on that from a scientific point of view, but the question was raised here, so there is not consensus about this. And we have to be sure that all agree on that. So, and I'm quite sure that we can get consensus or agreement for mutations, but not for DNA lesions. And you see what I mean? Yeah. So we have to work on that first. Yeah. And then we have to agree what is really adverse and what can be used as a marker or a biomarker. And EFSA has a cross-cutting working group at this stage to define what are biomarkers or markers to be used for risk assessments. And maybe we have to bring this area also in this group.
Yeah. Okay, of course, sure.
It's a little bit, we should be careful of the level of confidence that we ask for before we put something into regulation. If you think back about 500 years when ICH was created, and the older people amongst us may remember that, there was not a full agreement that these should be the three assays that are the best. But all of a sudden, you know, you had a host of assays, and different people were, of course, they were convinced that their exotic test would be the best. Nevertheless, everyone was forced to do exactly those 3, 4, whatever assays, and with time, the data grew, and you know, the confidence grew, and also the understanding that this is the right way. And later on, they were adapted. The revision 2011, when we said, hey, why don't we integrate the things into in vivo and all that? So it's not always when we make a regulation, when we put them down in words, in a regulatory document, that we do that from a position that everyone in the world is 100% convinced that's the right thing. No, we do it often from a position of, yeah, could work, and let's see where that goes.
Well, that is, yeah, just let me jump in and a bit back to what Frans Verstraete said. And if I got you right, your argument was like, okay, if we want to start a regulatory process on this issue, we need to have scientific consensus, right? Yeah. And Tanja Schwedler said, yeah, then we will never get started because we can never achieve this. No. How about you two? No. I mean, how do you judge this situation? Yeah.
Maybe to retract out a little bit from where we were deep down on the what tests, how tests, to come back up a little bit. I think most important thing, because I heard also we ought to be flexible, which is for a regulator a challenging position. You know, we want to give regulatory certainty while providing regulatory flexibility. But in terms of the tests and what it means, I think what we should be able to agree on a scientific basis is hazard identification, hazard characterization across the different fields. And then it comes to risk assessment, which is very different, because I hear my colleague here coming from the pharmaceutical field, and of course I used to work at EMA for 16 years. There, yes, you do the genotoxicity testing, etc., but you can always weigh it up in the overall benefit-risk balance. For food, you don't have that. For food, it's all about not having a risk. Also,
would you say that
chemicals, it's different.
You do have to have a scientific consensus before start regulation?
I don't think so, no. I don't think you need full overall scientific consensus. What you need to do is you need to have a basis in your regulatory field to move forward. We haven't even achieved that from what I heard. As EFSA, we've sent 6 people here and had every workshop, we had colleagues in there reporting back, and with a specific task of saying, do we have consensus on a way forward? We also, as EFSA, have 35 million euros to spend on the way forward in various areas, not only on genotoxicity, and we're willing to do that, to put the money where our mouth is. It's something that other EU agencies cannot do. It's certainly something we can do, but we want to have the way forward. And at the moment, what I heard was whole genome sequencing, NAMs, other areas where we have exotic testing, you called it. I'd like a consensus on the way forward. We don't need a full consensus on this is where we, you know, everybody agrees that this is the ultimate test because we're looking for the impossible to achieve. It doesn't exist. If that existed, we would all go for it.
Okay, Frans Verstraeten.
Well, yeah, as I said, there was a bit of a discussion of this scientific consensus, but what I could indeed see as a way forward is indeed let us work out this improved or this new or improved assessment of genotoxicity, work it out within, I would say, with the contaminants where there are no regulatory requirements, and where you can see if that would indeed, if indeed the confidence grows within indeed for the contaminants on this new way of assessing genotoxicity, that could then be a step up to indeed change the regulations for regulated products. While indeed for contaminants now there are no regulatory requirements, so it is up to the scientific panel to come to a scientifically based outcome as regards genotoxicity, and that will be then accepted by the risk manager, because we have no requirements from a legal point of view how the assessment, the risk assessment itself for contaminants has to be performed. And so by doing this, that can bring in more confidence, and indeed in a later phase, it could then also have consequences for the regulated products and have consequences for the
Nope. I think we lost it.
Yes, unfortunately you're frozen at the moment, so we can't hear you anymore, and your picture got stuck. We'll try to fix this, but I would like to give one aspect.
Confidence.
Oh, there you are again. Okay, we lost your last sentence, unfortunately. You were frozen a bit. Yeah. Can you repeat your last two sentences?
Yeah. So that is what I say, is not that it is a scientific consensus, but by indeed having that, for instance, for contaminants where we have no regulatory requirements, and by working in this new way of assessing genotoxicity, that indeed works out, and as regulators we accept, as risk managers for contaminants we accept that, and by doing this, if there is indeed, let us say, this is an outcome of a discussion, of a scientific discussion within the contam panel, and indeed over time that there is indeed more in the scientific world a growing confidence on this approach, that could then be a step up to indeed also have consequences for the regulated products and for the related regulatory requirements. Thank you.
Thank you very much. I would like to get back to one point that you mentioned. If I got you right, you said, like, why not try to just do things? Try to think about things we can already do and implement them, and then let's see where they take us. Did I get you right?
Yeah, a little bit to that. Not so, what I was after was, you know, probably we'll never have 100% consensus on that, and that, you know, let's define whether on which points we have enough consensus to say, okay, let's try to put that down in some legislation in a way, or whatever you want to call that, in some guidance document, and then work from there. Because the other advantage of a guidance document is that people do similar things, and you start comparing apples with apples. And then work from there. And as I said, a guidance document is never, you know, made for eternity. It can always be adapted and whatever, you know, modified, or even, you know, put to an end. We had a number of OECD guidelines that we put to an end because we said those tests are no longer used on, or they are not informative. You know, that all exists. So.
But then, let me ask you back: is there something that you have got after these days? You've got a strong feeling like, yes, we do all have a consensus here. Is there something that you would say, like, yes?
Well, not all. Again, as I said, we probably never end up with a 100% all consensus. But what I meant is this benchmark dose modeling and the uncertainty factor type of thing, and then get to factors, and of course maybe to be defined what is a level where I have an acceptable risk on a level of genotoxicity, or whatever, you know, these type of things. But use quantitative parameters and measures to work with those, and not just this black and white.
What about the others? What do you feel? What is the consensus within all of us here? The thing that we could start from?
Yes, I think that talking about scientific consensus is a bit aspirational, I think, because I think that scientists, they tend to debate and to enter into confrontation because this is science. I mean, science is made of uncertainties, and how to handle uncertainties, it is not a fixed rule, and we have to accept this. But it is true that I see a trend on certain things that appear to be a majority. I've been hearing a lot of support for the BMD approach compared to other approaches, and it is one of the things that I come back home because in ECA we are considering both. I would like that the BMD, BMDL is considered with more attention, of course, when you have the data to do it. But I can recognize that gradually there is a trend on certain things. But to talk about consensus, I think it's a bit aspirational because it does not belong to a symposium. I think.
Well, I think we have consensus that more work is needed. That's the easy one. You know, there is more research needed, more work is needed. Okay, we have consensus on that. Benchmark dose modeling, we don't even have full consensus because it was actually something EFSA published last year, and not even there's complete consensus on whether the one we are proposing is the right one. So even there it's difficult. But for sure, having more information on quantitative methods and assessing that and having it validated, I think everybody agrees on that. As soon as we have an appropriate validation of a technique, regulators are delighted to use that in a weight of evidence approach, and then risk managers, I don't know whether they're delighted, but are certainly willing to then adopt that. So yes, there's consensus. We just all hear that it's going to be a decade. But it shows that there's a lot of work to do.
Thank you very much.
I also think we have consensus in the fact that we have to prepare our risk communication, because when we are there that we can use the quantitative approaches, we have to be also able to communicate this. That is a challenge that we have to start also quite early. So,
and there, if I may,
sure.
Immediate response. Because there's the biggest difference between the pharmaceutical industry and food. You have the risk communication on the package leaflet, and you tell the patient all your chances of adverse effects. We're not doing that for food. When you get your pizza, for all ingredients, get a package leaflet. Imagine that. Therefore, the reason why in food we are maybe a bit more risk averse than in the medical area, in the medicines area.
Thank you. Professor Dogliotti, did you observe any points where you saw general agreement or disagreement amongst the participants?
Okay, so first of all, I mean, when I was saying flexibility, I was referring to the scientific community, not to regulators. I know that you are not flexible. No, I'm joking. I'm joking. No. So,
we try to be within reason.
No, I was thinking about an example where mechanistic evidence entered into the decision tree. There is an example that I think is quite interesting, that is the identification of human carcinogens. Because I think that we have to face the fact that the number of studies of chronic in animals, or chronic studies, are going to decrease. I mean, also for consideration about animal welfare. So we have to accept the idea that the mechanistic evidence should have a weight in the evaluation of, in that case, for IARC is identification of carcinogens. For us, it might be evaluation of genotoxic compounds. And talking about, you said, Tanya, mutation, okay, but then what about DNA damage? So I think that what they did at IARC was to find a panel of key characteristics that describe the mode of action of carcinogens, and they took it from the literature. So, and then at that point, you have really a broad array that helps in this context very much, because it reinforces your conclusion. So genotoxic agents have multiple way of action. And so I think that maybe we could take that one as a possible example of how to proceed.
But is there something that you would say, I saw a general agreement on this point, or maybe even a consensus about an aberration? What is to be avoided, definitely? What didn't work?
Unfortunately, we have weak data about correlation of genotoxic effects with adverse health outcomes. This is a problem because I think it's mainly because of poor standardization of the genotoxicity assay. We cannot make a correlation between the two yet. But again, I think that I know that there are projects also within OECD to have AOP together with genotoxicity assays. This might be a great help because there the outcome is the adverse health outcome. So this will help a lot for this correlation between the initial event and the adverse health outcome.
Thank you. One thing that I realized during this talks this morning was that a lot of you said, before you start risk assessment, you need to grab all the data from all the data sources you can probably get and collect what you've got, and then you can choose your approach. And every once in a while, someone said, there might be data, but we don't get it, or we don't have it. So do we need more data exchange? I remember one, I don't know who it was, but one said, maybe industry has it, but we don't.
Yes, I can try to address these. The possibility to exchange data is important. In the spirit of one substance, one assessment, we would like that when you address one substance and you address this substance under different regulation, you have the same data package in order to get to the same conclusion, at least on the hazard, for example, where you don't see the need to have a declination because of the specific application. The data exchange has been impeded to a good extent by legal boundaries because data are submitted normally by company for the use under a certain regulatory framework. There are confidentiality issues, intellectual property right, and it has been only under certain circumstances that a consideration of data under different regulation could have been easily performed. We have now a proposal from the Commission that we call in jargon data regulation, where we anticipate the possibility to build a data platform where all the data will be collected, and the possibility of reuse these data that will be in the data platform under different regulation. I think that this would be a major step forward in this direction. So it is acknowledged. It is something that does not allow to apply the all available data principle as we would like. All available data should be that if there are relevant data about the substance that can instruct the good assessment of this data for the safe use of this substance, it should be considered. And at the moment, it is an objective, but there are some impediments to get there. It will be greatly facilitated if this proposal will be then supported in the regulatory field.
Thank you very much. Is there a general need for more collaboration, by the way? No, Mrs. If you want to add something.
Yeah, I wanted to take that one.
Okay. Yeah. If you like, does anybody see a need for more collaboration between the different fields involved? Or
for me, sorry, for me that's a no-brainer because, of course, like data, to go quickly back to data maybe, again, a no-brainer that, yes, we need more data, but it doesn't solve the problem. It just means we have more data to look at the evidence that we had for titanium dioxide, now silicon dioxide for BPA, what regulatory agencies spend here, also in getting all the data together, is enormous nowadays. But without that, of course, you're lacking part of the assessment that you wish to do. So yes, of course, but if we believe more data, more studies will necessarily lead to a fixed point of, again, going back to the binary genotoxic or not genotoxic, we are wrong. And I think everybody recognizes that here. That what at best you can do is then do, and that's why the BMD modeling comes in, etc., is try and come to a tolerable level. And that's where it's become so difficult also with risk managers, because then it's in what field are you in and what is tolerable, and there it becomes very difficult. Regarding collaboration, it's partnerships in terms of assessment is a future necessity, because whether it's in pesticides, in feed, in food, we do not have the resources alone anymore. We need to also prioritize. EFSA, when it was built in 2002, was built as a independent agency. We're now talking about interdependencies, because without BFR, without ANSES, without Health Canada, without FDA, US EPA, etc., we will not achieve the level of data integration and information in order to make these decisions at the right level.
I see you nodding, so you would agree on that?
Yes, I agree, of course. But also, I think that it's quite important also to keep sort of, I mean, this meeting might be a sort of golden standard in the sense that I think that to have really people from different disciplines was extremely important. So I think really also to improve, to move ahead, it's very important not to have just a single voice from a single angle, but to be really together. I really think, I mean, really congratulations, it was a very good meeting, I think.
So do you think we have everybody aboard, or is someone missing here? Is there some voice that you would like to integrate next conference?
I don't know, I mean, I think that we were quite covered for different disciplines, right? So at least, I mean, in our workshop, this was the case, right, Kevin? I mean, we had so many.
If you could do us a favor and talk into the microphone so everybody can hear you.
No, I think that we were quite well covered there for the necessity also of understanding better what is going on. And so, I don't know, Tanya, what you think?
Well, the field of genotoxicity was always quite open-minded to not only leave all this or keep all disciplines on board, but also all stakeholders. And I feel what we learned here at the conference is that especially the colleagues from industry are quite advanced with respect to the toolbox. So, and even so, risk assessment should always be independent. This still allows us to take the scientific ideas of industry on board. And I feel we really should do so when we are going towards a quantitative approach, because we can all agree that we would like to have this quantitative approach. And we all agree without compromising on human health and without compromising on safety of food, feeds, drugs, chemicals, products. Did I forget someone? I don't know. Yeah. Everything. But we have to have all stakeholders on board. This does not necessarily mean that they influence the risk assessment approach or drive the risk assessment approach, but we have to take into account the science which is located in this area, because it's very advanced in the field of genotox.
So do you feel like the network is already fixed, or do you feel like there need to be structures implemented to get together at a different level?
No, I feel that workshops like this, they work out fine. So we have to prepare. Whenever we prepare to work on the guidances, I feel maybe it's too early to state industry should be on board when we develop guidances. That's a bit too harsh on the European level, first of all, I would say. But we have to clarify that the scientists which are involved that develop the new guidances, that they are fully informed and know what happens in industry and what is the scientific knowledge out there.
Hans-Jörg Martens, let me ask you, is there a special need at the side of industry? Do you have special requirements or wishes?
For what?
For this whole process or for collaboration?
Yeah. I have to say, you know, I have more information, of course, insight into the drug industry, and there, this sharing, what we said before, is, I don't think it's a major problem. As I said, we don't develop genotoxic drugs; we throw them away early in the process. And you know, as I said, it could be a waste of a resource of chemical space that we can't tap into because we can't define a safe level of using them. So otherwise we don't have them. It's a bit a pity because you also you will not get information on genotoxicity versus carcinogenicity because you only put non-genotoxic compounds through carcinogenicity studies. As I said, it's a bit. But otherwise, no, I don't see that as such a big problem. What I also hear, that sharing, that this would, you know, turn the whole thing upside down and fix things in one go. I don't think so. And the other thing is what we just discussed is that involvement of the different. I think that's still special. As you say, you know, in the meetings you're very open and you know it yourself in those workshops, be it in the national or international societies, or be it in other whatever, you know, HESI, IWGT. They're usually very broad, and we also have that sort of tendency to invite people from different, how should I say, from different groups, like from industry, from academia, and from the regulatory bodies together. Nevertheless, mostly, if you talk about genotoxicity, it's genotoxicity people from these different. So that's special here, and that's also something where you guys should be congratulated, that you have people here that are not genotoxicity people, but come from the risk assessment, and so we learn about a lot about that. So I found that quite remarkable, and yeah.
Would you like to add something?
No, just to complement, indeed, we have been discussing about data sharing, but I was far from considering that this was a solution. It is just one element of the system. And the concept of one substance one assessment go much beyond that. It concerns also the alignment of methodologies. And I think, as it was said already here, symposium like this, consensus like this, where you see the growing of methodologies and potential proposal in the perspective of different regulation, it's also an opportunity for an harmonized way how certain things can be picked up. Because if you have the same regulatory question at the end, and you have the same science, and you have the same data, you expect that there is consistency in what is discussed. And in the area of the hazard assessment, where, for example, harmonized classification is playing a purpose, clearly this is an expectation. So I think that the consensus like this is also an opportunity to see that if the regulatory question is the same, there is basically a mutual consideration of what is the technique, what is the methodology that has a favor, a consensus, and we have an alignment of methodologies where maybe historically, for some reason, they have been developing a bit differently. But in the eyes of a scientist, it doesn't make sense. In the eye of a regulator, neither.
So if you say this is a nice opportunity to pick up on some things, what is it that you would personally like to pick up on after leaving here? Or whom?
I mean, it's a good question. I think it was interesting for me to see the same question, how it is addressed in the food, for contaminants, in the pharma, in the REACH, in biocide, And indeed, I try to look the things also in analytical terms. There are some elements that appear to be more mature and having scientific favor. And I think that these elements should be considered when we are updating our guidance, etc. We have to look for a progressive alignment in that sense. So.
Thank you very much. Let me take a quick look at the audience. Yes, we do have a comment. There should be a microphone somewhere around you. And please press the button so that we can hear you.
Hi, this is Sophie McKenzie. I just wanted to go back to Franz's comments earlier about getting a scientific consensus. Certainly for the workshop 2 we were in, a lot of the failure to reach consensus was because of hesitation, I'll say, from the regulatory representatives or representatives from the regulatory agencies. What I would say, and what I challenge, is in order to shift the regulatory paradigm, we need the key questions that need to be addressed from the regulators. And then, because we already seen on Monday that, you know, from Paul White and others, that a lot of the information is already out there. If we have the key questions that the regulators need in order to shift the paradigm, then we can start to answer those questions and move the needle on this.
Okay, so let's pass that one on. Let's pass it directly to Brussels. What is the key question that you have? What is your need? What would you like to have? Which answers do you need?
Well, what are the key questions? That's difficult because, of course, we have, I would say, different approaches for the regulated products compared to the contaminants. In the contaminants, it is, for me, the key question is, as long as indeed, based upon the approach that the panel, now talk in EFSA terms, if the panel can conclude as regards indeed the concern for public health related to a certain substance, be it a genotoxic compound or not in food, that would be for us as a regulator sufficient, that indeed there could be that. And as I said, from that point onwards, if you see that this works for the contaminants and there is a growing confidence in such an approach, then this could then also be indeed have consequences for the regulated products and the regulatory requirements. So that I would see like this, because okay, I think it is upfront very difficult to change regulatory requirements if there is no experience gained with a certain approach, and so that's I think also a key point on that. Thank you.
Thank you very much. Let me pass your question on to our panel here. Are there any questions that you can identify where you say, like, that's what we need? If we got the answer for that, we can move on. Helpless views, I can tell you.
Having seen that workshop, yeah, exactly. Put the hot potato in my lap. No, having seen that out of the workshop, there's not even agreement on the usefulness and future of the Ames test. That was, for me, a revelation this morning on something that is really not extremely recent. Answering such key questions becomes a ping-pong because, okay, we can all retreat to our safe space, which is, the key question is it safe? So the key question is it genotoxic or not? So the key question is, what test do we need to show whether it's genotoxic or not? And then it's the key question, which test do we accept? Well, when we have the test agreed, here all of you are experts, much more than some of us, let's say, I speak for myself, all of you might be experts, than some of us. And what I hear is not an agreement whether it's on the quantitative testing, which one is exactly validated, how do you get to that curve, how do you get to the end point, or it's on the binary testing or in vivo testing, where we say, no, I even wrote down whether, you know, not capable of reaching the decisions. What at EFSA we do, and there we are maybe a little bit different in the sense, is that we take all the evidence of scientific literature also into account on top of what we have to come to the best possible scientific opinion and output. And that allows, in some cases, to take really into account also non-strictly regulated, mandated tests to see how they influence our decision. Okay, but that doesn't help you to say, oh, we want one test. We want one key question, one test. That doesn't exist. That's what I said in my beginning statement. If it did, it would have been 5 minutes. Discussion over. Thank you very much. We all go home.
Thank you very much. Any more questions? Yes, please. Can someone pass the microphone, please? There should be one. Oh, thank you so much. Thanks for the service.
Yeah, thank you very much. Yeah, and thank you for the insightful discussion. So we heard in work group 3 that the low-hanging fruit is potency, and it was in the context of potency within groups of associated compounds. And actually that was also discussed in work group 4, which I was a part of. And it was in terms of potency, perhaps even between compounds in different structural groups. So there appears to be a kind of emerging consensus on what is the low-hanging fruit. And we've just heard around the table here about, you know, what are the problems and the things that stop us moving forward. Is it worth to focus on the potential things that can be done as a part, instead of the potential problems? And what can we do now to move forward in terms of starting to implement perhaps approaches that are on a step towards the ultimate goal of quantitative risk assessment, and perhaps be able to articulate around relative potency as the next steps on the process, and what would be, you know, the way to start moving forward in that way?
Thank you very much. Who wants to respond?
Sorry, from my maybe ignorant point of view, you know, the Kruger-Dunning effect is not applicable to me. I'm not an expert, but validation.
Anybody else who wants to respond? Yes. Could you? Yeah.
So, the potency. I mean, I think that the main problem, at least, I mean, I do not have really a deep experience on that, but when we tried also within the scientific opinion of EFSA to really grade the chemicals, so to have a feeling of the potency, the problem is that the quality of the assays that were run sometimes is so poor that you cannot do anything. So I'm insisting the standardization and the validation of the assays is very important. I mean, it's very important that before doing an experiment, we think what we are going to do is going to be useful, or just, I mean, nobody will consider this data because they are not reliable. So I think that this is an important.
I do not fully agree on that. That's a low-hanging fruit, so to say. It's a low-hanging fruit maybe for a lot of endpoints, but not for genotoxicity. How can we agree on a potency in the area of genotoxicity if we cannot quantify genotoxicity? So is it less or more likely less genotoxic or non-genotoxic, or maybe 100% more or less non-genotoxic? That I do not see that's a low-hanging fruit. I completely agree that in vitro approaches, in silico approaches are extremely helpful, but first we have to agree on the issue of genotoxicity if we want to quantify. Otherwise, I cannot see how it can be a low-hanging fruit.
You took the microphone back. Do you want to respond something? No? No?
No, just to say thank you very much. It was very helpful.
Thank you. Thank you. Do we have any questions or remarks in the chat? No, not yet. Okay. Oh, wait, yeah. 1, 2. Yeah, you can start. Yes, please.
Ooh.
Yeah, just following on around the point about consensus, so scientific consensus. We haven't defined what it means. Some people are saying absolute consensus. I would say there's not even absolute consensus that the Earth is a sphere. So these definitions are...
Oh. Just, yeah, change. Thank you.
Didn't like my controversial comment. So here we go. So what is scientific consensus? According to definitions in the dictionary, a generally accepted opinion or a general agreement. So based on that, within our workshop 2, there was that. So less than 10% of people might have disagreed. Generally, there was acceptance. So if we go with true definitions, there was consensus, there was scientific consensus. So these really need to be defined previously rather than having 10 different types of definitions. Any comment?
Thank you very much. 1, 2.
I'd like to comment on 2 different questions. The first one is validation. I always hear validation. I'd like to hear what is the data set that should we validate against? What's the gold standard? That's question 1. And the second is a comment on the current genotoxicity assessment that you're applying. Yeah? If you perform an Ames test in 5 strains, and let's assume in 1 strain the number of revertant goes up to 1.8-fold over controls, then that's a negative, right? We don't do anything. We don't regulate that substance. If it's going up to 2.1-fold, it's a positive. We do everything to avoid exposure to that substance. That's the point where quantitative would make sense, to release this dichotomy.
But let me get to the first point of your question. Is there something like a gold standard that you can validate against?
No, there isn't, at least not in terms of one test system. You know, you went back to that and say, hey, since whatever, 40 years we're doing the Ames test, and now people come up and say it's irrelevant. Of course, that's very frustrating for all of us who've done that for the most part of their career. But, you know, it's in the data. And it's something what I meant before, right? All people do the Ames test because it's sort of considered one of the key pillars of doing genotoxicity assessment. And so you're only getting to these type of data to assess them if people do that. That's what I meant initially. We have to define this level of agreement, of confidence, of scientific, whatever, you know, consensus that we have to say, this is now good enough. We never have the 100%. This is good enough to put something down in a more formal way and then work from there and then come back to it and see, has it worked? Has it misled us? How often? I mean, there's no other way of doing things, right?
Thank you. We have another question.
Just to expand on what George was saying, we've had this debate in the past. What's the difference between consensus and unanimity? I mean, do you have to be unanimous or is consensus sufficient? But aside from that question, I was just going to comment on collaboration and data sharing. I don't think anyone would disagree that that's essential, but there's different types of collaborations. So as a research scientist, it's really easy for me to collaborate with George and Andreas, but when I go back home and interact with regulators, sometimes the regulators think, you know, well, that's just a bunch of eggheads playing with numbers. So we need to get close to the regulators, and I thank the BFR for bringing us close to the regulatory community. And I'd have to thank my bosses, too, at Health Canada. They basically told me, you need to do research in support of regulatory decision-making. And here's the regulators. If you don't talk to them, we won't give you any funding. So we're going to put you in this room. We're going to close the door. We'll give you some food, but you can't come out until you come to some path forward that's good for both of you. So I think that's essential, those kinds of collaborations, not necessarily between George and Andreas and I, but the interactions with those who make the regulatory decisions.
Thank you very much. So you get to have white smoke then? Or how do they know that you came to a solution? Okay, any more questions or comments? Not so far. Yes, please.
Just one, because potency came up, and I think that statement actually, at least in our work group, of course, was in the context of very defined situations. Like, for example, I mean, we are working on pyrrolizidine alkaloids, the BFI is working on them, a lot of other people are working on them, and we see the same potency ranking between different groups. So wouldn't that be an example? These are unavoidable plant-based impurities. Wouldn't that be an example where such a system could be of use? Or PAHs, or maybe now nitrosamines, because there's a lot of work going into that. So when you know already the mode of action, they are DNA reactive, you know everything you need to know. So why not start with those? So I think it's possible. I mean, and then just the Ames test, because that came up. I mean, that wasn't, we weren't expecting any consensus on that. And that's not surprising because it's in every guideline, right? So if you tell people, well, tomorrow we don't like that data anymore, then of course you don't get consensus on that. I think it was brought in as a thought starter. I mean, is there a need for modernizing the standard battery? Is there, because there might be better approaches. That was all. We didn't expect consensus. So, and then my last comment is in the context of quantitative approaches, especially in the in vivo space. And since I didn't hear that from Paul, I just wanted to say, or from George, or from Andreas, I mean, we use the International Workshop on Genotox testing, which is a tripartite approach. And I mean, there's a lot of work going on there. There's really, the concept is bringing people in from all geographies and regulatory agencies and so on, and that's what's happening. But there's certain, it's always very difficult, to be quite frank, to get people involved that come from European agencies, and we hear the people are not allowed to come. And then I think it's easy to present it as non-consensus. So just, sorry, that's a bit controversial, I know, but I needed to say that.
Okay, so let me ask you this. Do you judge it the same way? Is it hard for people from EU regulatory boards to attend meetings with scientists? And if so, what can be done against it?
Two things I want to say. First, I think EFSA is very proactive, and no, it's not that hard, but let's be careful what they are, whether they are scientific conferences or industry-led events, because we are always extremely careful about the independence, and that is also necessary and asked for by the risk managers. But I wanted to come back also to the collaboration issue, because collaboration, we find, you know, now this word goes around, I'm sorry that I'm using it as well now, but it's not only about collaboration, it's about co-creation. Rather than EFSA doing a guideline and BFR sends an observer or ECHA sends an observer, doesn't work anymore. We need to get together and try and co-create guidance in the appropriate space in order to really, for everybody to have the trust and feel it and represent it. Because otherwise, as a regulatory agency, we often find ourselves, yes, this is the EFSA guidance, and then people tell, this is the EFSA guidance, but BFR does it slightly differently because we also have a guidance, and obviously that's not helpful to anybody. So it's something where really, that's a paradigm shift for us regulators to try and achieve, that we do things truly together. I talked about that in terms of the interdependency rather than independence. We go to interdependence with other regulators so that we come up with those guidance documents that you find useful, that answer key questions, but for everybody. Because otherwise you find yourself in this regulatory, I wouldn't call it jungle, but in this regulatory space where it depends really exactly always what agency you're talking to. And that, I think, we need to, for two reasons, we need to sort of get over, A, in our own interest, because only if we are together can we, with all the data explosion that we have, can we address this and the complexity, and B, it will help in the future for clarity, because we need to, as regulators, continue to be also a help of innovation in the EU. I talk now as an EU regulator.
Thank you. Do you want to add something from ECHA's point of view?
No, just to say that I think that regulators, at least if I consider the reality of European agencies, are quite permeable. I don't have the feeling that we avoid discussion with the scientific community. I would say that there are not so many events like these where we have science, which is not biased, but clearly posing relevant question for the regulators. You might have big scientific conferences where the discussion goes in all direction, and you pick little things, or you might have some events that are very biased because they respond to a specific appetite. I think that event like this, without prejudgment, goes in a different direction. And as Nick was saying, it is a dialogue. I start saying in this panel that for me the question was what we want to achieve, because this is what a regulator will start with. And what I've listened in these days is more what science can bring at the moment. Very good, but the question is how it can fit in the regulatory framework, and what is the added value. And I think that the dialogue should be as such. It is not what can be picked up at the regulatory framework, but what really serves the regulatory framework. And sorry, regulation has some principle. The regulation needs to work. Yes, you can have some situation where you have a dichotomy, as Andrea was saying, that a threshold is exceeded and bring you in a different status. Yeah, I mean, it is how regulation works. It is how the speed limit works. And so the level of flexibility is impeded by the need of a regulation to work in a certain way. There is no bad or good regulation. It is a regulation that works, a regulation that doesn't work. And I think that there are some principles that need to be respected. And one of the principles is regulatory certainty, and legally certainty for companies in the first place. So they need to know what are the rules and how they can comply with the rules. And if something becomes very fluid and ad hoc, this would be very difficult, not only for authority, but for the company and the applicant in the first place. So, yeah, I think that there is the need of this dialogue. Maybe there is the need of more events like this. Thank you.
Thank you very much. You wanted to add something?
Yeah, I wanted to add something because Franz could not be with us in the morning, and there was a little elephant in the room, so to say, in the morning. So there was workshop 1, and in workshop 1 that addresses genotoxic carcinogen, there was a statement from the workshop participants that they could go for maybe a quantitative approach, or they could discuss about the MOE if there would be a question answered. That is very central. And it was more or less related that this is more risk management question, and maybe this we should discuss. The question was, what is the acceptable level of risk with respect to cancer incidence? So this is something, sorry, but we have to discuss.
So, do you have an answer for Tanja Schwerdle?
No, I don't have a straightforward answer. Of course, it's always, of course, we have to, certainly in contaminants, we do accept a certain risk, because we do not, for a lot of contaminants, we get indeed, even after regulation, we have indeed MOEs which are still far below, for genotoxic carcinogens, far below 10,000. So we cannot say, we cannot, even with regulation, even if we for the outside world, for easiness, we say the levels are safe. They do in reality not full guarantee safety, because we are not reaching an MOE of above 10,000, which would be of low concern. But on the other hand, we also do not know what is exactly the risk. Is it now indeed 1 in 100,000, 1 in a million extra cancers that we have following that? This is and it is of course, we cannot escape this discussion. I understand that, but it's not easy, because of course we say we accept 1 in 100,000 or 1 in a million. I still remember my first days in the Commission when indeed in WTO there was indeed an attack on our aflatoxin regulation, which was considered to be too strict, and on which indeed EFSA still now says that it is still of a health concern, the presence of aflatoxins in the EU. And yeah, there was this study of the World Health Organization which says, okay, our EU legislation saves two lives in a million, or even in a higher billion, and that was attacked, and my director phoned me then from Geneva, and he said, yeah, is that true? Well, I said, yes. Well, I cannot say it is true. It's true that it is in the study. Well, he says, yeah, okay, if there are at least 2 out of a million, that's for me a sufficient basis to defend our legislation, because I don't want to be one of the 2. So this is indeed a difficult discussion, if you accept saying, okay, we are 1 out of 100,000, 1 out of a million. So, but of course, if we go to quantitative assessment, possibly we might have to be able to determine the level of risk. But at this moment, I do not want to pronounce what would be an acceptable level of risk in that sense. Thank you.
Does anybody else in this hall or on the podium has a strong opinion? What is an acceptable risk? No, I think, oh, there is one. Some brave one.
I don't have a clear number on that, but I imagine that within the name of European Food Safety Authority, the word food should not only be restricted, and I assume that's not the case, to genotoxic substances or impurities or contaminants in food, but to food safety at all. What is safe foods? The level of sugar in soft drinks causes possibly hundreds of dead people in a million, and there is no legislation advancing in this direction, and we accept that, right? The general public drinks Coke and drinks sugary stuff, and I eat unhealthy food and stuff. Nobody cares, but we have a lot of concern for contaminants. Just food for thought.
But does that automatically mean that there is no use in searching for this acceptable level, or no? Okay, anybody else who wants to? Yes, please.
I think we're probably going to end up in a situation not with one acceptable level, but with as many acceptable levels as there are reasons for or use case of intake. The acceptable level in pharmaceuticals with a very defined benefit is probably going to be higher than the acceptable levels in food. And where does every other regulatory environment fit between maybe those 2 numbers? Maybe there's somewhere we need to be even stricter than food or less strict than pharmaceuticals. I don't know. But each set of risk management and risk assessors are going to need to come to their own decision about what is an acceptable risk in the context of the exposure through the market they regulate.
Okay, so the ball's back on your side of the playing field.
Maybe to complement a bit what we have been saying about acceptable level of risk, I think it is not a question that can be put in isolation. When we are saying that we have an acceptable level of risk of 1 on 1 million, well, we say that basically we use this benchmark in an assessment because it appeared to be conservative enough. Of course, we might give the false illusion that we have such a predictive capacity, but I don't think that we expect that 1 person over 1 million will be affected. there is an assessment which has been based on a series of conservative assumptions, etc., and there will be these elements that is needed to make it work and complement. And of course, these elements need to be considered in terms of impact, in terms of workability of the legislation, in terms of level of protection that it gives, to take it as a figure of person that will be affected and sacrificed to the cause. It can be misleading. In that sense, yes, in some cases, the need of the risk manager to understand what is the level of protection is important and might be the missing element. But it needs to be put in the context. It is not just a question which is extrapolated. Okay, do you think that 1 person over 1 million is low enough? No.
Yeah, that's
it's
basically
never enough.
what meant.
But it can work in a certain regulation. So indeed, once the system is built up and there is a clarity of the uncertainty that we have, and we are able to quantify in a certain level this uncertainty, and to understand what is the impact of this figure once it is put there, it is for the risk manager to understand what can be the figure that work well.
Okay. I don't see any more questions. So I would love to come back to the subtitle of this conference, which is Challenges and Future Perspectives. And I would like to invite you back onto our panel, Frans Verstraete. From your point of view, what is the most urgent thing to tackle now within the next, let's say, months? Let's not put it to 10 years. What needs to be done and who needs to do it?
Well, that's also a challenge to answer for me. I would indeed, as I also indeed mentioned in my introductory talk, and I apologize that I'm a bit maybe biased towards contaminants, but yeah, I would like to, well, as I said, for a number of contaminants, genotoxic compounds, which we have regulated, we are still indeed in a situation where even if they are regulated, we have still MOEs far below 10,000. And it would be for us as a risk manager to see what is further needed. To have indeed a quantification, what does it mean really as regards the additional risk on cancers or the fact that we are not at a situation where would be considered of a low concern. And that would be helpful because, okay, we accept that we are now at an MOE for acrylamide at 300, 400, for other contaminants even lower. But what does it mean for indeed the real quantification of the risk in order to define maybe much harsher measures if needed to, that is, I think, would be already very helpful if that could be realized on the short term. Thank you.
Okay, thank you. Tanja Schwedle.
Frans, that's really helpful, and I fully agree the contaminants is the easiest area to do so. But just to remind you, at least in the EFSA content panel, we will not do so if there is no guidance, because this will be criticized. So the first step would be to work on the guidance, because otherwise, you know, this won't work.
Okay, any other opinion on the point, what's the most urgent thing to solve within the next 10 weeks? Let's make it shorter.
Well, within the next 10 weeks, it's publish the recommendations and the outcome of the workshop, right?
It will be done, by the way. If anybody wants to review or rewrite or reread some things, there will be a documentary of this conference within the next 10 weeks, let's put it like this, on the Academy's website.
Yeah, I would agree with that, because that's why we also came in force from EFSA to really look at the outcomes from the workshops where we can do something such as the margin of exposure approach, etc. But it's not going to happen within 10 weeks. That's for sure.
It's just because you said you didn't want to wait for 10 years because you've got the appointment with the plumber then.
There's something in between 10 weeks and 10 years.
Okay, let's make it 10 months. What do you think, what should happen within the next year? What's the most urgent thing to do, and who needs to do it?
I think Tanya said it. The work on the guidance from where we, and discuss also, but work on the guidance where we can bring forward improvements on the application of a quantitative risk assessment and margin of exposure.
Anybody, anything else on the wish list? Yes.
Yes, this is a wish. I think that would be very important to have a much quicker transfer of scientific innovative tools, but also scientific new concept in the field to people who are doing risk assessment or regulation. Because, I mean, the example of Health Canada and also NIHES, Susan, I mean, that I see there, I have the feeling that in Europe we are going much slower for this type of integration. You go much faster. There is no way. So I was wondering whether NIHES, for instance, is it working in close collaboration with EPA? Because there are documents that I read, for instance, from EPA, which are very much science-based. Maybe they say very clearly that they are not meant to be regulation, but there is already a message that is transferred from the scientific community to the risk assessors. And so, and this Health Canada is another, I think, very good example. So what is not working here? Why we are so slow in this integration? Maybe this is very unpopular, what I said.
Thank you very much. So, yeah, we've got rather 10 months to go until Christmas. Anybody else? Something that they wish for? Yes, 1, 2.
A bit of a technical question. What I wish for is some guidance about how to use endpoint data where it's not mutation or chromosomal abnormality. I mean, I think people in this room know that I'm definitely enthusiastic about the quantitative use of data for mutation and chromosomal abnormality for risk assessment. But it gets a little more complicated when you're dealing with transient effects like strand breaks or gamma H2AX foci or things like that. And that's what the NAMs use. So how are we going to use those endpoints for quantitative analysis and risk assessment of genotoxic substances? Then it's really genotoxic. I mean, I deal with mutagenic, not genotoxic. So for genotoxic, where do we need to go? And how are we going to get there?
Thank you very much. Anybody wants to respond something? Otherwise, I'll ask you for your contribution.
I mean, those will not be the core tests for foreseeable future. They will be additional tests, and it's like with any new methodologies, essentially the confidence in them grows when you have more data. So it's really, there's no trigger to push to say we get from here to there. But you know, people need to use them. People need to publish their experience. So others can see that, and others can try to recapitulate that, and then you can also use them in a regulatory context as additional evidence in a scientific argumentation about a case, this and that.
But Hans-Jörg, this is true for your area. This is not true for the contaminants field, because in the contaminants field we will live forever with these old tests, because there is no money out there to do these studies. So we have to really answer this question.
Yeah, but there is no way around getting data on new tests, on new approaches, and publishing them, really scientifically working on, hey, this is a new approach, and what does it tell us?
But we will have the daily problem: should we use the comet data or the micronuclear data or not? And there are no studies submitted to an agency in the area of contaminants. So this is why I agree with Franz. We can start with the contaminants, but in the area of contaminants, we have the most poor data set. So it's not true for the pyrrolizidine alkaloids or nitrosamines, but for most of the others, So we can start with some examples, but then it will get complicated because we cannot decide on what others do, which type of studies. There is no regulation for what you have to do in the field of contaminants.
So what could be done to improve the data set in that field?
I don't know.
Anybody, any idea? Not yet. Something to work on within the next 10 months.
Maybe we need AOPs that connect those endpoints to adverse outcomes. I mean, in a quantitative manner. So that would provide the foundation for using those endpoints in a quantitative way for risk assessment, and with a convincing argument that they're indicative of something bad happening.
You have a lot of answer to our questions, Paul. You should be there quite often with us.
Okay, any more questions or comments? How about the, yes? Online community, just give me a quick sign, yes, no? Yeah, we do have, so you go first and then we go into the chat.
Thank you. When I'm thinking as an assessor, I would like to see the connection between the effect that is measured and the adversity, and even if there are different key events in between, one should have the knowledge, the quantitative knowledge as well on the key events. And if further testing is managed with in vivo data, of course the route of exposures have to be taken into account, and the dose ranges that might be also expected in human populations and subpopulations have to be respected in the test design. But as I've seen from a Nature paper from 2 years ago, I think it was, there were tested also compounds that interact directly to the DNA, and they have, of course it was not in vivo but an in vitro approach, but it seems quite logical how they did that. And it was like 20 compounds were tested several times with the same test system, and only 3 of 20 gave the same mutation signature in sequencing. So for me it would be wonderful to see, but it is very expensive, of course, with a relevant and somehow predictive exposure design, let's say this way. What is the mutation signatures, maybe first in an in vivo transgenic animal experiment? What are the reproducible mutation signatures that I get out from different control substances from which I know they are doing frame shifts, base mutations, nucleate excisions, insertions and deletion mutations, chromosome breakages. All this mode of action should be somehow in a reference experiment being comparatively shown that they give similar mutation signatures in the target organs with a relevant exposure scenario. So that is a call for way more research? Of course, this is very expensive, but if we want to, if the scientific community wants that regulators are starting to think about acceptance of the quantitative approaches, we have to know which is what is the correlation between the mutation signatures and the adverse effect, whatever, must not be cancer, can be other kind of maybe cytotoxic events that may follow. But this is what is needed to also establish a relevant AOP on genotoxicity. And I know by chance that there is also OECD struggle on having a genotoxicity network of AOPs, which means that wants to interlink different AOPs and see where are the knots and where are the bifurcation points, and to study the possibilities how cells can react to putative mutagenic compounds. It's a real big endeavor, and I see that with the modeling, we see technically some data sets can be very well modeled, and but this real cause effect relationship cannot anticipate it from several well-done examples. We need to have the different genotoxic endpoints linked to either mutation frequencies or mutation signatures. And the problem is that you have also the kinetics inside. You have to know how much of the compound comes into this target cell or organ. So a lot of thinking before, as has mentioned before, has to be done before such an experiment is planned. So it's just some ideas where we see the conundrums or the hindrances maybe before we can really draft a guidance, because how can I be sure that mutation frequencies for one gold standard compound in one target tissue is the same in the other target tissue?
Does everybody agree, or is there a dissent on this? Yes, who wants to answer?
I can neither agree nor disagree here, because I understand, we are all aware of the fact that genotoxicity, mutagenicity is a very, very emotional endpoint also. But sorry to say, also in the regulatory field, we can never ever be 100% sure. I completely agree. From a scientific point of view, it's important to have all this information, but we cannot wait 10 more years. At a certain point, we have to make some decisions. Once again, we should absolutely be sure that this will not compromise on human health, will not compromise on the safety of food, feeds, drugs, chemicals, and products. But we can also do this by factors. We do this in so many approaches. And why not with Genotox? We cannot wait for the next 10, 15 years. Even if we give Paul all the money, he will take 5 to 6 years to answer this. We can't. This will take forever.
Thank you very much. We got questions and remarks from the chat, I learned.
Yes, the first question is, do you think that we could use pharmaceutical data to develop an overview of AOPs so that it can apply it for risk assessment, for instance, of unintentionally added substances?
Did you get it? It was a bit hard to hear.
Not 100%. Could we use information from pharmaceutical molecules to develop AOPs? Yeah, of course. Yes. Was that the answer you needed? I mean, why, or the other way around, why should it not work? Of course, if you have information. As I said, usually we don't use and characterize genotoxic compounds very much. We try to get rid of them under the current paradigm. But otherwise, yeah, you can do that. And you can do that like with any other molecule.
Okay, thank you. Any more questions? Yes, please.
So there is another question that goes into a similar direction what we have discussed already in the morning session. And the participant agrees totally that quantitative approaches for genotoxic chemicals are needed for substances that cannot be avoided. But on the other hand, when dealing with regulated products, don't you think that performing such quantitative risk assessment for a single chemical won't be enough to achieve that level of protection, and that you would need to take into account human exposure to all the other known sources of exposure to genotoxic substances? So it's a long question.
That's essentially what you do with these quantitative approaches that were introduced here. So you get a benchmark dose, and then you work with uncertainty factors, right? And you use, for example, an uncertainty factor if human exposure is unknown. If human exposure is known, for example, I don't know, for example, for a drug, right? You could say you shrink that uncertainty factor. You could shrink it to 1 if you're absolutely sure that it's the exposure that people will have and nothing else, right? So what was the question again?
So I think it's important to discuss other known sources of exposure of genotoxic substances, and if the exposure of other substances is really known. of other potentially
so
genotoxic
you're
substances.
talking about additive effects or combination effects.
That's what I understand from this question.
Yeah, but of course you could do that. I'm not really familiar with the regulation, how that is done, but different ways of thinking it. If you think whatever they act by a comparable mode of action or are repaired by by the same pathway, you could say let's add them up. Other things. Yeah. So it's not it's doable. Yeah.
Thank you. I would quickly come back to something that you just said, and you said it very emphasized. We can't wait for another 10 more years. We need a decision now. We need to start making a decision. Which decision is the one that you are most longing for?
Well, maybe I did not state this correctly. I'm not searching for the decision right now, how to do this, but not to stop and hide behind the fact that we have to wait for the research data. Well, we should start now to work on the guidance. We should bring together people in the field that have the ideas. Maybe then people might identify and say, we need exactly this or that research, and we heard from EFSA there's money for this. This is what I heard. Sorry, but I feel we cannot wait. I'm quite sure it will take a while. And I was frustrated when I heard, about 10 years or so. I would be more optimistic, and I heard from other voices, like from Diane, that the MOE approach, if we start working on that, we can be there in 1 year or so. This is very feasible, and this will help already the management. And towards the quantitative approaches, why not give it a try for the contaminants? And I feel this is also what the question was asking. Maybe people might accept the quantitative approach for the contaminants because they are not avoidable. They are out there. But they might not be ready to accept this for regulated products like pesticides or food additives. And maybe it's good to give it a try with the contaminants and let people know, look, it's better than to do nothing. And then we can decide at a later stage, maybe in 5 to 10 years, how to do this with the regulated areas.
So if you look into those phases, you have the consensus that was missed earlier this day. Everybody was nodding. You want to add something?
Yes, I do. I mean, it takes 2 to tango, and it takes at least 4, 5, 6, 7 to collaborate, co-create on guidance. So I fully agree on the guidance. And I saw in one workshop there was this suggestion of EMA, ECHA, EFSA, OECD. I would include BFR, I would include ANSES, I would include DTU, RIVM, big regulatory agencies. Let's focus here. This was the focus on the EU. To get together and try and agree on some of the answers to these questions. That's number one. And then, as somebody mentioned very kindly, biomarkers of effect. We have let's put it this way, a working group, international working group for drafting a guidance. And because we want to talk here also to the FDA, Health Canada, US EPA, and it's incredibly complex. Doesn't mean we don't go for it. We will go for it, but the backgrounds are so different that there we might actually take a decade, I think, for that particular. But for EU agencies and member states agencies that have the resources to be involved in that, and also the interest and the budget, I heard money, also BFR has money, no? Then absolutely to get together and also agree, yes, that in some areas we can fund projects, as we do.
Thank you very much. Okay, please.
I just want to react on the strong remark that we shouldn't start with pesticides, and because that's already safe, but because it's a binary thing, yes or no, it could also be that some pesticides are now regulated as not being genotoxic, while actually they are. So if it seems to work quite well, then maybe it's also making pesticides more safe. That's just.
Thank you very much. Any more comments or questions, or bright ideas that you don't want to take home for yourself, but share them with us? Yes, please. The bright idea, the solution, there he is.
Well, it's not necessarily a bright idea, but I guess it's a question really for the sort of regulators and regulatory authorities. Because obviously one of the consensus that we seem to have reached is that it would be really good to have the quantitative approaches available. And that sort of change can only really happen with scientific creativity. And so we've got some examples where we have to be creative in the contaminant space. We've also had regulatory pressure giving those of us working in the cosmetic space the necessity to actually try these new approaches and see what can be done in that space. So what can regulators and regulatory authorities do to fuel that creativity? Because you're not going to get an answer, fully formed answer, without having that creative phase first.
Okay, thank you. Does anybody want to comment on that?
I gave the answer already. Let me give it again. We have 35 million that we're putting into research every year. And NUMS is a big, for example, new approach methodologies is one of the big areas. Also PERA, so pesticides, environmental risk assessment, etc. We have programs that we invite research institutes to apply to in order to move science forward. I need to also defend my colleagues from other agencies. Not every agency has that possibility of grants. These are real grants, they're not procurements, and that is where it's really helpful.
There is also the PARC initiative. So it's not ECA that is putting the money, but it is research, yeah, that we are partner, and it is exactly research that should feed the regulatory needs. So it is an area where this can fuel innovation in that sense.
So we've got the means, we've got the manpower, we should make progress, I guess. If there are no more questions, oh yeah, there is one. Can we pass a microphone to Christine, please?
Yeah, thank you very much. If I had to wish, then I would wish more conferences like this, but being more focused and filtering a little bit more. Because for me, it was rather a kick-off meeting where many different challenges and problems were discussed, which is very important, for sure. But I think now we should start to ask on specific questions so that we have a workshop with all necessary persons that should be involved, scientists and also regulators, but not so many persons at all. Maybe one specific person from one specific party who's really willing to do something and to work on something, having a case study, answering this question, publishing this question, and coming then to the next question.
So what is the most necessary question for you? Just share it with us and everybody can take it home and think about it already.
I have to think about it because I noted indeed a lot of questions from all the workshops, but I think a general consensus was that the BMD approach is the most promising approach for quantitative assessment, and we should now go on with this. Or another focus could be also the contaminants, because this seems to be the most promising field to enter the discussion or sustain the discussion on quantitative risk assessment. So I think these are two consensus that we can work on.
Okay, thank you very much. Any more questions? No. So let me end this discussion with a huge thank you